Only after 2 iterations, we realized that colonic concentrations of chemicals like polyfluoroalkyl substances, PFAS, and melamine can actually match or exceed plasma level. That's a crucial detail when we're trying to connect lab results to what's happening in people's bodies.
To put numbers on that, studies have found micromolar concentrations of PFAS in human blood and urine, and even in dairy products. For example, the CDC reports that nearly every American has detectable PFAS in their system, which suggests chronic, low-level exposure is the norm rather than the exception.
But if those levels are widespread, why aren't we seeing more obvious health effects? Is it possible that the gut microbiota changes are subtle, or that we're missing long-term impacts because they're hard to track in large populations?
Here's the takeaway. The challenge is bridging that gap between what we see in vitro, like bacterial inhibition, and the complex reality of human exposure. Regulatory limits often lag behind new science, and vulnerable groups, especially those in agricultural or polluted regions, might be at higher risk.
There's also the issue of cumulative exposure. People aren't just exposed to one chemical at a time, they're getting a cocktail from food, water, and even household dust. That makes it tough to isolate which compounds are driving changes in the gut microbiome or health outcomes.
Could better public health monitoring help, or are we limited by the fact that most exposure assessments rely on snapshots like a single blood or urine test rather than continuous tracking?
Continuous monitoring would be ideal, but it's rarely feasible at scale. For now, the best we can do is push for more comprehensive biomonitoring and advocate for stricter regulatory thresholds, especially for chemicals that show microbiome disruption at levels people are actually exposed to. Only after 2 iterations, we realized that the same efflux pumps bacteria use to survive pollutants like closantel are also giving them a leg up against antibiotics. Here's the takeaway. Resistance isn't just about one threat at a time. These pathways are multitasking.
That's right. The AKRR locus and RND-Tol-C efflux pumps, for example, are highly conserved across gut species. In the study, mutations in these regulators didn't just help bacteria survive chemical pollutants, they also increased resistance to ciprofloxacin, which is a clinically relevant antibiotic.
But if these efflux pumps are so central, why aren't we seeing more direct interventions targeting them? Is it because blocking them could disrupt beneficial functions in the microbiome as well?
Exactly. Efflux pumps aren't just defense mechanisms, they're involved in normal bacterial metabolism and homeostasis. Inhibiting them could have unintended consequences, like impairing the bacteria's ability to process nutrients or interact with the host.
And there's another layer. The study found that loss-of-function mutations in metabolic enzymes, like those for branched-chain fatty acid biosynthesis, were also selected under pollutant exposure. That suggests pollutants could be reshaping the metabolic landscape of the gut, not just resistance profiles.
So, if pollutants are driving both antibiotic resistance and metabolic shifts, does that mean people exposed to more environmental chemicals might have a higher risk of gut-related diseases or less effective antibiotic treatments?
There's a strong possibility. The co-selection for resistance and metabolic changes could compromise both the resilience and the beneficial functions of the microbiome. For instance, altered fatty acid biosynthesis might impact gut barrier integrity or immune modulation.
Here's what stands out. Chemical exposures in our environment aren't just a background risk, they're actively shaping how our gut bacteria evolve, sometimes in ways that undermine both our health and our medical tools. What surprised me most was just how many everyday chemicals, things like flame retardants and plasticizers, turned out to have strong antibacterial effects on gut microbes. We used to think their impact was limited to their intended targets, but this new research really challenges that assumption.
The numbers are pretty striking. Out of more than 1,000 pollutants screened, researchers found 588 inhibitory interactions involving 168 chemicals. That's nearly 1 in 6 substances showing some kind of antibacterial activity, and about 30% of fungicides and industrial chemicals had a measurable effect on gut bacteria.
But are we sure these effects are actually relevant for people? If these chemicals only show activity in vitro, can we really say they're disrupting the microbiome in real-world conditions, or is that still uncertain?
Here's the takeaway. Even if the findings are from lab tests, the sheer scale of interactions, especially with chemicals like closantel, tetrabromobisphenol A, and bisphenol AF, raises real questions about our current safety assessments. These compounds inhibited more than 1/3 of the gut species tested, which suggests a much broader biological reach than regulators have considered.
And it's not just about the number of chemicals. The study also found that certain gut bacteria are especially vulnerable. while others seem resistant. For example, some beneficial species that help with digestion or immune function were among the most sensitive. That could have downstream effects on health if those populations are suppressed.
If that's the case, why haven't toxicology studies picked up on these side effects before? Is it because they focus on acute toxicity? Or are there gaps in how we evaluate chronic, low-dose exposures?
Most safety assessments still focus on direct toxicity to humans, not on collateral effects to the microbiome. This research highlights a blind spot. Chemicals designed for one purpose can have unintended consequences for gut health, and that's something regulators and scientists need to address going forward. Only after 2 iterations, we realized that the models trained on pesticides just couldn't predict the effects of pharmaceutical drugs accurately. Here's the takeaway. Chemical classes really matter when it comes to machine learning predictions in toxicology.
That's a crucial point. The dataset included over 1,000 chemicals and their interactions with gut bacteria. But when random forest classifiers were trained on one group, say, pesticides, the accuracy dropped sharply if you tried to apply them to pharmaceuticals. It's a clear sign that the underlying molecular features differ substantially between these categories.
If that's the case, what's stopping researchers from just building separate models for each chemical class? Wouldn't that sidestep the generalization problem?
It helps, but there's a trade-off. Building class-specific models means you need enough high-quality, standardized data for each group. Right now, the lack of harmonized datasets is a bottleneck, especially for less studied chemicals.
Here's where computational modeling could really accelerate chemical safety assessment. If we can standardize data collection and reporting, these machine learning tools could flag risky compounds early in the design phase, moving us closer to safe-by-design chemicals.
But how realistic is it to expect regulatory agencies to adopt these models? There's always the concern about transparency and interpretability, especially with deep learning approaches.
Regulatory uptake will depend on validation. For example, random forest models offer some interpretability by ranking molecular features, but deep learning embeddings are harder to unpack. Agencies will likely require rigorous benchmarking against experimental results before integrating these predictions into policy.
The momentum is there, though. As more standardized datasets become available, and as models are validated across chemical classes, machine learning could become a core part of chemical risk assessment frameworks.
Only after 2 iterations we realized that chemicals considered safe for humans could still disrupt gut bacteria in ways regulators never anticipated.
If we start requiring anti-gut bacterial activity as part of hazard evaluation, how do we handle the sheer volume of chemicals already in circulation? That seems almost unmanageable.
Here's the takeaway. Integrating microbiome testing isn't just about new chemicals. It means revisiting legacy compounds and updating risk profiles, which could reshape entire product categories.
A 2023 review found that over 60% of common food additives have never been screened for microbiome impact, so the scale of unknowns is significant.
But even with advanced gut-on-chip models, can we really capture the complexity of individual microbiomes? Or are we just adding another layer of uncertainty?
We'll need a tiered approach, starting with high-throughput in silico screens to flag risks, then moving to targeted lab assays. That way, we prioritize resources without getting paralyzed by complexity.
And policy-wise, this could mean new labeling requirements or even reformulation mandates for products shown to disrupt beneficial bacteria.
If the field embraces microbiome-aware toxicology, we're not just protecting gut health, we're redefining what chemical safety means for the next generation. Thanks for joining us as we rethink the boundaries of toxicology.